Pediatric high-grade gliomas are very aggressive brain tumors that are responsible for the most cancer-related deaths in children. Over the last 25 years, there have been no effective therapies developed. One of the main reasons for this is current therapies are great at targeting the properties of tumor cells, but they fail to target the properties of other cell types within the tumor. Now, our lab has shown that up to 30 to 40% of all the cells within an adult high-grade glioma are composed of tumor-associated macrophages. Macrophages are like the garbage men of the body. They go around cleaning up debris and dead cells and clearing infections. We do not usually see these cells in a normal brain. However, in the case of a high-grade glioma, we see a massive accumulation of these cells coming from the blood. And instead of doing their job in killing the tumor cells, they actually promote tumor growth. Now, everything we know about these cells comes from studies in adults. We know virtually nothing about them in a pediatric setting. How did these cells get to the tumor? What did they do once they're inside the tumor? And lastly, how can we therapeutically target these cells to prolong patient survival? That is what my PhD is on. And to answer those questions, I'm using human patient samples and genetically engineered mouse models. And what we have found so far is that in both mice and humans, those tumors that have the highest levels of macrophages are the most aggressive, and they have the shortest survival. We see that those tumors with high levels of macrophages have their own unique genetic code that allows them to produce high levels of chemoattractant proteins called chemokines. These are signals given off by the tumor cells that attract these cells from the blood into the tissue. We see that the macrophages then respond to these tumor signals by producing their own chemokines, thus creating this very aggressive loop where the tumor cells attract the macrophages then respond by attracting even more macrophages into the tumor, thus fueling tumor growth and malignancy. Now, lastly, what we are currently working on is identifying the specific mechanisms or the chemokine most responsible for this macrophage attraction to the tumor. Because if we can do that, we can then develop specific targeted therapies aimed at preventing these macrophages from moving into the tumor and thus prolonging survival in this very dismal disease. Thank you.